Kami umumkan kepada adik-adik mahasiswa untuk bisa menempati bangku yang di depan. Karena acara kita akan segera kita mulai, kami silakan kepada adik-adik mahasiswa yang di belakang untuk bisa menduduki bangku yang berada di sebelah depan, karena acara kita akan segera kita mulai. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera buat kita semua. Yang terhormat, Dekan Fakultas Kedokteran Universitas Andalas. Yang kami hormati, Wakil Dekan Fakultas Kedokteran Universitas Andalas. Profesor Dr. Kedokteran, PhD, selaku narasumber kita pada pagi hari ini. Ketua Program Studi S1 Biomedis Fakultas Kedokteran Universitas Andalas. Bapak Ibu tim SOTK, tim GKM, Fakultas Kedokteran Universitas Andalas. Mahasiswa Program Studi S1 Biomedis Fakultas Kedokteran Universitas Andalas. Bapak Ibu para tamu undangan yang saya hormati. Puji dan syukur kehadirat Tuhan Yang Maha Esa, berkah rahmat dan hidayahnya, kita dapat berkumpul bersama pada acara Kuliah Tamu World Class University, Program Studi Ilmu Biomedis, Program Sarjana, Fakultas Kedokteran Universitas Andalas. Tak lupa beriring salam kita kirimkan juga kepada Nabi besar kita Muhammad SAW. Dengan mengucapkan Bismillahirrahmanirrahim, acara Kuliah Tamu World Class University, Program Studi Ilmu Biomedis, Program Sarjana, Fakultas Kedokteran Universitas Andalas, hari ini Kamis 18 Mei 2023 dimulai. Mengawali acara ini, kata sambutan dan laporan dari Ketua Program Studi S1 Biomedis kepada Ibu Dr. Desi Arisanti, MSc, kami persilahkan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan selamat pagi semuanya Bapak dan Ibu. Pertama kali teman-teman yang terhormat kepada Bapak Dekan Fakultas Kedokteran Universitas Andalas, yang terhormat Ibu Wakil Dekan 1 Fakultas Kedokteran Universitas Andalas, yang terhormat Bapak Wakil Dekan 2 Fakultas Kedokteran Universitas Andalas, dan tidak lupa kepada speaker kita pada hari ini yang terhormat kepada Prof. Dr. Kedokteran PhD dari Universitas Putra Malaysia. Terima kasih atas kehadiran dari Bapak dan Ibu, dan juga terima kasih kepada panitia dan pengelola daripada program studi Universitas Andalas yang mengadakan kegiatan daripada salah satu cabang World Class University. Kegiatan ini merupakan bagian dari kegiatan World Class University yang diadakan oleh Universitas Andalas. Dan hari ini merupakan kegiatan puncak daripada rangkaian kegiatan invited lecture yang diadakan di Prodi S1 Ilmu Biomedis. Nah, sebelumnya kegiatan ini juga ada dalam bentuk teaching class yang dihadiri oleh mahasiswa S1 Ilmu Biomedis, mahasiswa S2 Biomedis, dan mahasiswa S3 Biomedis dengan topik-topik mata kuliah tertentu. Kemudian kita juga dalam rangkaian kegiatan ini, kita juga ada praktikum yang dipandu oleh invited lecture kita, yaitu adalah Prof. Queen. Selanjutnya juga di dalam agenda invited lecture ini kita juga melakukan FGD terkait dengan rencana riset kolaborasi ke depannya dan apa-apa peluang yang akan dilaksanakan. Nah, sekarang ini adalah merupakan general lecture atau kuliah umum yang audiensnya adalah semua mahasiswa dari Fakultas Kedokteran dan juga dosen-dosen dan juga dari universitas yang berada di luar daripada Universitas Andalas. Semoga kegiatan ini memberikan manfaat bagi kita semua dan ini akan lebih mendukung kepada program daripada Universitas Andalas untuk menjadi universitas yang bertaraf internasional atau World Class University. Demikianlah laporan daripada kegiatan ini. Saya kembalikan kepada MC. Assalamualaikum warahmatullahi wabarakatuh.
Kata sambutan sekaligus pembukaan oleh Bapak Dekan Fakultas Kedokteran Universitas Andalas. Kepada Bapak Dr. Afriwadi SH SPK UMA, kami persilakan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Asyhadu an la ilaha illallah wahdahu la syarika lah wa asyhadu anna Muhammadan abduhu wa rasuluh la nabiyya ba'dah. Segala puji dan puja kita menajatkan kepada Allah Subhanahu wa taala atas berkat dan rahmat Allah pada kesempatan pagi ini. Alhamdulillah Allah telah memberikan kemudahan kepada kita untuk dapat bertemu dalam rangka untuk melaksanakan salah satu kegiatan akademik, yakni kuliah tamu yang inisiasi dalam upaya kita untuk mendapatkan World Class University. Itu memang topik utamanya, tapi dengan demikian kita berharap kolaborasi ataupun apa yang kita upayakan untuk menjadi World Class University tersebut, setidak-tidaknya kita dari Fakultas Andalas, terutama adalah Ilmu Biomedik, itu memberikan kontribusi yang positif terhadap upaya-upaya kita untuk pencapaian Universitas Andalas yang lebih baik lagi. Amin, amin, amin. Yang saya hormati narasumber kita kali ini, Prof. Queen. Good morning, Prof. Queen. Thank you for your here now. Selanjutnya kami ucapkan salam penghormatan kepada Kaprodi Ilmu Biomedik, Kaprodi S2 Biomedik dan Kaprodi S3 Biomedik. Ya, walaupun kemarin kita berhasil Kaprodi, ini barangkali mudah-mudahan apa yang kita pilih pada kemarin itu dapat bermanfaat. Amin. Dan yang kami cintai para peserta ataupun para mahasiswa atau peserta didik di lingkungan ilmu biomedik. Kemarin kita sudah sepakat untuk menjadikan semua daripada peserta didik kita itu dapat mengikuti perkuliahan ini. Nah, perkuliahan ini memang mudah-mudahan akan mendapatkan arti penting bagi kita semua, sehingga kita mendapatkan share keilmuan yang bermanfaat dan keterampilan yang memadai yang akan di-sharekan oleh narasumber kita, yaitu Prof. Queen. Mudah-mudahan saja kita semua bisa menjalankan kegiatan ini dengan baik. Dan tentu kepada semua peserta kami berharap mari kita ikuti kegiatan ini dan mudah-mudahan segala daya serap penyerapan yang kita ambil dari kegiatan kali ini mudah-mudahan sesuai dan sampai tujuan apa yang kita harapkan. Itu adalah harapan-harapan kita. Kemudian tentu juga pada saat ini kami juga mengucapkan terima kasih atasnya kepada Prof. Queen yang telah berkenan dan berbagi waktunya dengan kesibukan beliau untuk hadir di tengah-tengah kita secara offline pada kesempatan ini. Tentu kami ucapkan terima kasih besar. Semoga amal ibadah dibalas di atas kuasa. Dan kami juga mengucapkan terima kasih kepada Kaprodi, Wakil Dekan dan semua kita yang terlibat di dalam proses kegiatan ini, terutama kepada program-program tercetus dari Direktur Pembelajaran. Mudah-mudahan ini akan menjadikan UNAND akan lebih jaya di masa-masa yang akan datang. Akhirnya mengucapkan Bismillahirrahmanirrahim. Cara kuliah kamu dan narasumber mungkin pada pagi ini kita buka dengan respon. Semoga Allah meridhai dan berikan kemudahan, berikan kemanfaatan yang lebih terhadap acara yang kita lakukan dan kita ajak pada hari ini. Demikianlah yang dapat kami sampaikan. Kepada Allah kita minta ampun dan semanusia marilah kita saling maafkan. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Bapak Ibu tamu undangan yang saya hormati, untuk acara selanjutnya yaitu kuliah umum yang akan dipandu oleh moderator kita, Dr. Mutia Laleni, MSc, dengan narasumber kita, Profesor Dr. Chehyen Queen, PhD, yang merupakan Deputy Dean Faculty of Medicine and Health Science University, Putra Malaysia, Selangor, Malaysia. Kepada moderator kita, disilakan.
Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin. Allahumma sholli ala Muhammad wa ala ali Muhammad. Colleagues, staffs, and students who have been here today with us in our special programs, General Lecture, Bioinformatic Role in Drug Discovery with Professional Technologist, Dr. Hyuk Queen. Okay. Before we begin our program today, let me introduce you our honorable speakers for today. Professional Technologist, Dr. Hyuk Queen is the Deputy Dean, Graduate Studies, Industry and Community Relations and Income Generation, Faculty of Medicine and Health Sciences, University Putra, Malaysia. He is a Fellow of Academy of Science, a Registered Medical Laboratory Scientist, Malaysia, a Charter Scientist, Fellow of Institute of Biomedical Sciences, United Kingdom, a Chartered Biologist, Fellow of the Royal Society of Biology, UK, Fellow of Royal Society of Chemistry, UK. Professor Dr. Chia contributes immensely in non-governmental organizations as well. He holds the positions as the advisors for Malaysian Biomedical Science Associations, or MyBiomed, President for Biomed KL, and Korean Government Scholarship Alumni. Currently, Professor Dr. Shah is one of the Malaysia Allied Health Professor Councils. He is also a certified professional in virus assessment and biosecurity. Please give applause. Furthermore, Professor Dr. Shah is an established scientist with more than 200 scientific publications, 5 patents, copyrights, and won numerous awards in national and international research exhibitions. He has successfully led 15 grants in the area of molecular diagnostics, genetic engineering, drug discovery, molecular biology, molecular microbiology, medical biotechnology, molecular medicines, genetics, cancer biology, and bioinformatics. In 2017, Professor Dr. Shah was successfully awarded as Top Research Scientist Malaysia. Please give a round of applause for our speakers today. Without any further ado, please have our honorable speaker, Professor Dr. Shah today. Time is yours, please.
Thank you, Madam Chairperson, for the kind introduction. A very good morning, salam sejahtera, selamat pagi semua. yang berbahagia Prof. Dekan Fakulti Kedokteran, yang dihormati pegawai-pegawai daripada Fakulti Kedokteran, dan yang dikasihi pelajar-pelajar daripada Fakulti Kedokteran dari semua sesi. So saya rasa bersyukur kerana dapat menghadir dan berkongsi maklumat serta ilmu yang dapat saya sampaikan nanti. So terlebih dahulu terima kasih juga kepada Sekretariat kerana mengundang saya untuk event pada kali ini. So izinkan saya bentangkan presentation saya dalam dua bahasa kalau diizinkan. Terima kasih. So now, today's I would like to share some of the information about bioinformatics role in drug discovery. Sorry for this slight technical hiccups. Now, allow me to just face the screen. Boleh. Sorry again for this slight technical error. Now, allow me to share these particular important topics today on the bioinformatic roles in drug discovery. So now, a drug can be defined as a substance intended for use in diagnosis, cure, mitigation, treatment, or prevention of diseases, or as a component of a medication. So now, drug discovery is a process of discovering and designing drugs. And this process is a very important and involving analyzing the cause of the disease and finding ways to tackle them. So this process is very long and expensive that it might cost millions of dollars and take a dozen years, and the accuracy of identification of targets is not good enough, which in turn delays the processes. So as all we know that there are various ways and means in drug discovery. So with the advancement of a new era, bioinformatics is generally defined as the research, development, and application of computational tools and approaches for expanding the use of biological, medical, and health data, including those to acquire, store, organize, archive, analyze, and visualize such data. And this particular approach allows us to explore the cause of diseases at the molecular level, explain the phenomena of the diseases from the angle of the gene, and make use of computer techniques and enhance the accuracy of the results so as to reduce the cost and time. So now, obviously, every single of the scientists and researchers in the world are racing to find a perfect therapeutic drugs. So what does it mean that all these therapeutic drugs? The drugs is defined as all natural and artificial made, chemicals that produce desired effects in the living organism when administered by the most favored route. So either is oral, intraperitoneal, intramuscular, and etc. So what makes a chemical compound acting as a pharmaceutical active agent? So these are the three main components, which is high affinity towards the target, selectivity with respect to the target, and high bioavailability and low toxicity. So now let's look at some of the history way back, how drug discovered in the olden days. So the discovery of drugs historically has been serendipitous way back in 2001. For most of its history, drug discovery has been a product of trial and error. So all you know in the olden days where there were scripts of these ubat-ubatan, so penemuan drug tersebut adalah secara kebetulan. And most of the time dia dahulu orang-orang menemui ubat melalui percubaan dan kegagalan, and menerusi also observation daripada animal, okay, daripada di mana binatang-binatang menggunakan tumbuhan-tumbuhan sebagai ubat-ubatan. So long before the pharmaceutical industry existed, drugs were discovered by accident, and their uses passes down by verbal and written records. So all you know, some of the discoveries bukannya hanya sekadar just direct, but sometimes it's accidental. So out of the particular research, accidentally they found those particular compounds. So according to Ball in 2003, throughout history, people have found by trial and error which berries, roots, and barks could be used for medicinal purposes to alleviate symptoms of illnesses. So most of the time that in the olden days that orang ataupun batin, mereka melihat tumbuh-tumbuhan di melata supaya dia menggunakan ubat-ubatan tersebut ataupun herbal untuk mengatasi simptom-simptom daripada keadaan penyakit tersebut. So for example, willow bark, which contains salicylene, which used as a fever reducer. So dahulu kalanya tak ada cara ekstraksi compound di dalam tumbuh-tumbuhan ataupun herbal. Mereka menggunakan keseluruhan tumbuhan tersebut and campuran tumbuhan-tumbuhan ataupun herbal. So dengan campuran herbal tersebut terdapat toxicity yang terdapat di dalam tumbuhan tersebut. So it's not an ideal way of using those full herbal medicine. So let's look at the whole this particular flow where the drug discovery in the 50s and 60s. So this particular picture capture, the flow chart capture from the Ratty and Chris in 2001. So initially in the very olden days, there are random screening and also serendipity that leads to obtaining this lead optimization. And then they found the candidate, and then there's a full development and subsequently into the market. So however, this particular process improved along the years, and drug discovery in the 80s, you can see that there's a lead optimization candidate, and then there is exploratory development. There's extra component of the exploratory development, and then subsequently to full development and into the market subsequently. So what happened to the newest 90s and beyond? There are a lot more component which is arise before the identification or the lead optimization, which is high capacity screening, focus screening, target identification, target validation. That is all actually supports the high identification and lead identification, lead optimization before they end up finding the lead candidate. And subsequently, from the lead candidate, then they were subject to preclinical development and subsequently clinical phases 1, phases 2, phases 3 before they launch into the market. So prosesnya semakin panjang dalam penemuan drug sebelum dia boleh dipasarkan dalam persekitaran ataupun dalam global. So now this slide shows you some of the snapshot of the modern drug discovery, that the process of discovering and designing drugs, which includes target identification. So this is something similar like previous, this more pictorial, that identify diseases, identify, validate the pharmaceutical target, identify lead molecules, optimize lead molecules, and then subsequently subject to preclinicals, clinical trials, and then the approval and stipulation. The next slide shows you what are the component that involved in each particular area. So you look at the target identification in modern drug discovery. So it comprises of bioinformatics, cheminformatics, and also data mining. These are all possible because of the database, available database, and also the technology know-how. So of course, when you look at the target validation, so you will have all this protein-protein interaction, genetic network mapping, protein pathway mapping, protein assay. And then it was subject to lead identification with a high throughput screening, virtual high throughput screening, structure-based models, protein ligand models, microarray, and the list goes on. And subsequently, even in preclinical trials testing, you have this ADME and toxicity, and also biosimulation can be done via this clinical testing. And subsequently, there will be an FDA submission for the launch of the particular product. So this particular bioinformatics or the electronic infrastructures of the screening allows the particular processes to be more complete and also more thoroughly done. Okay. So what are the tools, okay, available in bioinformatics to assist the drug discovery? So the process of designing a new drug using bioinformatic tools have opened a new area of research. So in order to design a new drug, one needs to follow and abide to the certain path. So first, we need to identify the target disease, studying interesting compounds, detect the molecular basis for the disease, and refinement of the compounds, and also QSAR and also solubility molecules to look at whether the compounds developed is whether bioavailability well and also solubilized in certain contexts. And of course, then it was subject to drug testing. Okay, now let me share with you this particular component on bioinformatics, which is the structural bioinformatics in drug design. Of course, there are many means of approaches in particular bioinformatics on drug designs. It can be either sequence-based approaches or structural-based approaches. I'm just highlighting here one of the approaches generally by most particular researchers. So these are the structural bioinformatics. It can be used to examine first the drug targets, which is usually proteins here, and the binding of ligands based on the particular 3D structures, and then it will lead to rational drug designs. So now, by using these particular approaches, you are actually removing the guessing, try and error. So from this particular point and these approaches, you'll be able, dengan adanya simulasi tersebut, dia mengurangkan kaedah teka dan gagal. Okay, because most of the time, kebanyakan masa bila kita menggunakan cara penekaan, so kegagalan itu akan diikuti because it's not the proper way of actually designing the particular drugs. So with these particular approaches, not only just removing the errors and also removing the tries and error, but it also saves a lot of times, and also it also benefit to the researchers. So now let's look at the traditional methods of the drug discovery here, as I keep on mentioning. Now, in the olden days, that most of the people use the whole plant derived treatment, and sometimes that will lead to isolation of active compounds. So I think majority of us who comes from this background will also using this approach, which is a traditional drug design approach. Getting the plants, isolate the fraction, identify the compounds, and then subsequently do the testing. This is something that very common. So every single of us who doesn't have the knowledge of bioinformatics will also go through these particular processes. There's nothing wrong with these processes. It's just that it gives us a longer processes to go through what we want to hit. Okay. So of course, these particular techniques also have advantage over the particular true bioinformatics approaches, because these particular approaches allow us to actually have the field and also the starting material that we wanted to use, the sources of the starting material. Unlike in bioinformatics, through total true bioinformatics approaches, you started off with sequences and all these structures, you need to correlate that with a particular synthesis. So you need to understand by simulating or predicting the structures, okay, dengan andaian daripada software tersebut, kita perlu bekerjasama dengan para chemists, okay, pakar kimia, and also those people who are biochemists, okay, to develop that particular structure. Unlike here, you can start out with this material that we can see whether it's doable. So then from the synthesis compound, and then people will manipulate, okay? So we will manipulate the particular structures to get a better drug. Sometimes from the compound, you will understand by the biochemist or chemist, they will understand that whether these particular structures is solubles and not solubles, and what we call the backbones of this particular, just to identify the backbone of the particular compounds, and then you can do derivatives from there. So this is something very common nowadays, okay? So now, modern methods of drug discovery, what is the difference? The modern drug discovery begins with a disease rather than the treatment. So like I mentioned before, traditionally, we use that particular plant to treat. But here, you look at the disease. We can use the disease model to pinpoint relevant genetic or biological components. And then from there, you will see that what are the possible targets, okay? Because this particular component, of course, they will look at the diseases per se, and then what are the available database, okay? So now, this particular flowchart shows the modern drug discovery. From the diseases itself, you get the biological or the genetic targets, and then you will find a discovery of lead molecules, and then design an assay to measure the function of the target, and use assay to look for the modulators and the target function, and uses a particular electronic infrastructures to screen or do the high throughput screening, okay, HTS. So, and of course, from there, you will get these small molecule hits, and then from there, the molecule hits, you manipulate the structure to increase the potency, okay? And to look at the infinity constants and the concentration, for example, you decrease the infinity constant and etc. So from there, the optimization of lead molecule into candidate drug. So as I said again, from this lead molecule, then it was subject to the synthesis. Dia bukan just sekadar just design from the particular software, but you must understand that how do we synthesize, okay, that particular compounds or molecules. So, and that fulfillment the pharmaceutical properties in the area of potency, absorption, bioavailability, metabolism, and safety. And that will go through the testing, in vitro testing, and also subsequently will lead to clinical trials. So of course, when you look at it, with these particular approaches, you have a lot of option hereby derive that particular derivatives from the particular lead molecules. So now this particular slide shows you one of these lead optimization molecules you can done virtually using this software, okay? So it's an open source that you can actually analyze the particular molecules, and this allows you to see the properties of these molecules into deeper, okay? So now what are the interesting facts about drug discovery? So all we know, and every one of us understand, that 90% of the drugs entering clinical trials fails to make it to the markets. All this because of the toxicity, and also the average cost to bring a new drug to the market is estimated 70 million. So therefore, it is not an easy route, okay, to come up with a particular potential drugs for a treatment for particular diseases. It is interesting, and we need to understand that most drug candidates fails in the clinical trials. After many years of research, millions of dollars have been spent on them, okay? Because as you know that, because at the end, when they enter the clinical trials, maybe the drugs are not fit to a certain population. So because without a proper screening or this particular high threshold screening, then this particular drug will not be possible in the use of treatment of the diseases. And most failure because of the toxicity, as I mentioned, or problem with the metabolism. So excretion of these particular drugs once they enter our body. So the key characteristic for drugs are absorption, distribution, metabolism, excretion, and toxicity. And efficiency, in other words, bioavailability and bioactivities. How do we understand and change this bioavailability and bioactivity? It is by bioinformatic approaches or software, these screenings approaches. So although these properties are usually measured in the lab, they can be also predicted in advance by bioinformatics, such as this particular software. So by using this particular software approach or bioinformatics approach, you will be then remove the, as I said, the cost of using it to test on the toxicity in the in vitro or in vivo. Okay. So this slide shows you the impact of the structural bioinformatics on drug discovery. So it's from the genomics, and then we'll move into bioinformatics, structurals, cheminformatics, and then structure-based drug design, and then ADMET modeling. So it speeds up the key steps in drug design process by combining expert on bioinformatics, structural biology, and structure-based drug design. So these particular approaches allow us to understand better rather than you just use a particular compound and then test it on the animals and also the cell lines. So how to identify these particular targets, what we call druggable genome. Okay. So they are related druggable targets to diseases. So over 400 proteins has been used as a drug targets. So as you know, because the protein possesses structures, so it is important to understand the protein as a whole, not just using it, but you need to understand the various means of the motif of the protein, the proteins, and also the major targets of this particular connection of the protein to the particular genes. So how to assess them? Once a target is defined for the disease of interest, the structural bioinformatics can help to answer the questions here. Is this a druggable targets? So it's not all particular structures can be used as a drug. So because you need these particular component molecules to bind to a certain area of these particular cells or particular receptors in order for them to take action. Does it have a sequence or domain similar to the known targets? Does the targets have a site where a drug can bind and with appropriate affinity? So all this can be answerable to a particular structural bioinformatics. So what are the roles of this structural bioinformatics in drug discovery? Finding pocket of modeling, as I mentioned earlier, lead identification or similarity with known protein ligands or protein or ligands, chemical library designs, virtual screening, and also lead optimization. So with this all these possible area, you can actually develop a more efficient and also better drugs. So apart from that, I think many of us, even we didn't start off our design by structural or sequence. I think along the line, or the biochemists or those who are involved in the drug discovery, they're combining the traditional methods with the existing approaches, which is the docking. So I'm sure that everyone who are involved in the natural products, so I'm pretty sure that at the halfway of the drug discovery, they will also kind of include docking as part of their studies. And this docking is also part of the bioinformatics approaches. Docking is a method used to identify the fit between the receptor and a potential ligand. So the goal of a protein ligand docking is to predict the position and orientation of a ligand. So which is a small molecule that is bound to a protein receptor enzyme. So these listed here are some of the accession to all these protein ligand docking software. So some of the software are freely available, some you need to purchase. There's a license bound to these particular interactions networks. So now after this particular docking, and I'm sure that some of us may also expose these particular molecules to all these virtual high throughput screening, VHTS. So pharmaceutical companies are allowed searching for new leads to develop into drug compounds. Okay, one such method is through virtual high throughput screening. So in this virtual high throughput screening, protein targets are screened against database of small molecules compounds to see which molecules bind strongly to the targets. And this will save a lot of us our time to understand what are the particular possible reaction when this target enters to our body. So now I would like to share this publication, recent publication in the literature. So mentioning this is 2003, this year paper. So there are bioinformatic screening of colorectal cancer causing molecular signatures through gene expression profile to discover therapeutic targets and gene and candidates agents. So from this particular paper, it actually described that how bioinformatics helps in identifying all these particular therapeutic targets. You look at it, it sounds very scary. When you look at the graph here, even more complicated. But once you are into this screening, and you will see that it is wonderful to identify a very useful compounds and targets. So it started off with like gene ontologies and also identify key genes of it, and then there's a therapeutic targets, and then lead to a molecular docking, and then conceptualization, also collection of the molecules. So all these are part and parcel of the bioinformatics. It is impossible for me to actually explain every single of it. This particular slide to show you, it is important to understand various aspects of the particular bioinformatics into fitting the drug discovery component. Okay, now this is also a recent paper saying that identification of disease-related adaptation splice transcript in myeloma strategies to target this alteration by RNA-based therapeutics. So all these approaches are using bioinformatic mean to understand their targets and also the molecules that how they bind. So if you look at this, now is a trend, okay, I'm moving towards that era where bioinformatics really play an important role in drug discovery and also therapeutics. So now that's all about this general information. Perhaps maybe I can share part of our research to show you how our component included this bioinformatics section in our research. So now just to share with you that we didn't start with the sequence or the structural prediction. That is because it's a bit because we do not have a very strong chemist team together with us and also biochemist team. So we will also play a safer role where we'll start with our own particular compounds that we can harvest from. So what I can share today, one of our research is on this particular Biocytomonas hummi. These are very novel genus that we found, our group found in Antarctica. So it was first reported way back 5, 6, 7 years ago, and it's really new, it's novel, and we named this particular microbes according to the island that we found, the Pulau. So the Pulau near the Antarctica called Barentus. So that's why the genus actually uses this Barentus name, Barentus simonas. So to cut the story short here, so we have actually understand the structures of these microbes. However, there's a lot more to explore on these microbes. So we're trying to see what are the compounds secreted by the microbes. So of course, these microbes is actinobacteria, and it's all known that many of the actinobacteria actually produces a lot of useful compounds. So we uses these bioassay guided approaches. So we have this culture strong fermentation, extraction, and then cell line toxicity, and then furthermore look at the metabolomic studies. So you look at this particular chart here, is not all totally traditional approaches, where there is component that we uses the bioinformatics to help out on our screening, to make it more comprehensive, like metabolic studies, and also some others component on the structural derivatives component. So now this is just to show you the overview of what are the solvent that we use to understand the compounds. That I think this is very common to everyone single of us who are doing natural products studies, right? So different solvent, and then you want to find which solvent is the best, and subsequently you find the fraction, and then this is where the bioinformatics comes in, that you're looking at the fractions, and also then goes to the identification of the particular fractions to what particular structures or compound that is inside this particular fraction. Of course, many of us will stop here. Okay, I believe many of us will stop here, the fraction, and then they're tested, then they find, and then they conclude that fraction have these particular positive or negative effects on the particular cell lines, etc. So, and all you know that we shouldn't just stop here. We should go extend by looking at what are the components inside this fraction. So this is something that I want to show you, that this to highlight the fraction. These are all through the hardcore wet lab. Okay, so we perform the real-time analysis, real-time cell analysis to understand what type of cells that actually this particular compound or the fraction actually inhibits. So these are all various type of cancer cell lines. So just to cut the story short, because it's a bit lengthy, because I'm not going to describe all in details. Okay, now we also extend this study to understand the mode of the cell death. So we found that some of the fractions really useful. And then we will then proceed with all potential assay, the caspase activity assay, etc. Okay, so on which particular cell lines. And then we also venture into looking at this cancer metabolism. So this is where we try to understand. So the fraction itself not just remain as a fraction, ataupun sebagai part of the fraction. So we perform this, what we call phenotypic microarray profiling. So, and we get all these information from the PM plates. And that's here where, after this, we will get this metabolic fingerprinting, everything. So now, in a normal setting that were conventional approaches, you will see that through various experiment, you won't be able to actually profile or fingerprint this particular interaction without the help of bioinformatics. I will show you why. It is because if you just rely on the conventional approaches, you will only identify some of these particular metabolic reactions. However, with the bioinformatics approaches, okay, these are all the Wells reaction. I'm not going to describe to you because it's more details, technical information. So from that particular place, so we get all these kinetics. So from the kinetics, we were then able to understand, okay, what substrate that belongs to or react to these particular cells. So now the story doesn't stop here. So what most important here, from the particular substrate, then we use database integrations. We will then look at the CAT pathway, etc., to identify the pathway. So now this is something that we actually incorporated some of these minor bioinformatics searches to see their interaction and also deduce what the pathway that involved with this particular fraction. So now after we understand that, we will then search further what are the mode of action, okay, of all these particular fractions and also compounds. So then we will then understand that where this particular substrate, that this particular compound that involved in these particular studies. So, and from this, we will then subject it for particular compound, understanding the compound inside this fraction, which is the structural elucidation. This is done with the help of a chemist. So now we have all these particular compound being identified, confirmed by the chemist. And fair enough that this will be the one of the first few bacteria, actinobacteria, that produces this DKP, dipeptides, okay? So, and then from there, we understand that which particular peptides actually produces the most positive effects towards the cell lines against the cancer cell lines. And subsequently, these are the conclusion from that particular studies. Okay, so now we have these all phases here. The first phase, the cytotoxic effect of this particular YBH, the B-humis extract, was selected towards human breast cancer cells. So, and of course, then later we went into the phase 2, which is using the bioassay guided fractionation, and to understand the mode of cell death, which is then we understand that is this caspase 3 and 7 activities in MDA and B231. Okay. And also then we look at phenotypic microarray profiling on the phase 3. So to understand what are the particular substrate and component that uses by this particular microbes in terms of their particular fractions. So a total of 5 compounds were isolated. So cyclopyrrolithione appeared to be the most cytotoxic against the selective MCF7 cells. So, and we can declare that this will be our first report on the biological activities for the bihumine. So now this particular data was published then in this paper, American Journal of Analytical Sciences, to share with the group that there is diketopiperazine. Okay? So Actinobacteria psychotolerant bihumine sebagai satu sumber sebatian diketopiperazine. So now, what I'm trying to highlight here is there are various ways of identifying of this particular compound or synthesizing. So in some of the component when you start with all this structural based design, then you need to actually work very closely with the chemists to synthesize that particular compounds entirely. So, but however, when you use bioassay guided approaches and also fractionation and all these things, you will have the starting material in hand that you will know where to source this particular natural compounds from. Not only using the whole particular fractions, but a specific particular compounds that comes from there. So of course, from here, you will have this particular structure backbone, and you can then lead to this particular derivatives from there. So now we do have also other type of approaches in terms of non-organic approaches, which is using the metal compounds, which synthesized by the chemists. So I'm not going to describe all the details because due to time limits, because of the time, and then these are some of the papers that derive from those components that are also using the docking to understand the compounds, the novel compounds that is this DNA targeting nickel complex that with this testosterone thiosamicarbazone, which exhibits selective susceptibility towards human prostate cancer. So these are something that come from the synthetic. So the synthetic chemistry can be also one of the lead in the drug discovery and incorporation of the bioinformatics approaches. So we are not limited that just to the nickel. We also then venture into gold. Okay, again, that these are all started with the synthetic. It's not from the natural, but it's synthetic chemical. Okay, so these are all synthetic metal complexes that we tested against biological assay, and we found that some of these synthetic approaches need some of these bioinformatic approaches to understand the properties of that particular synthetic compounds. So we have screened various of it, and some of it are very successful in terms of giving us a positive effects on antibacterial and anti-cancers. So these are all some anti-cancers targeting on certain activation pathway, like nuclear factor kappa B, okay, and ubiquitination. So there are also some targeting on the gram-positive bacteria. So that's something that we can share that to allow you to see that by using these bioinformatic approaches, combining with all these existing mean of approaches, you can get more information out of those components. Okay. So now, what I can conclude from today's talk, the drug discovery process is long and expensive, and it started from a target identification. After that, validates the targets and verifies the drug candidates. Due to the limitation of throughput, accuracy, and cost, experimental techniques cannot be applied widely. Therefore, in recent time, the drug discovery process has shifted to bioinformatics approaches such as homology modeling, protein-ligand interactions, and virtual high-throughput screening, etc. So bioinformatic approaches has been a great importance to develop fast and accurate target identification and prediction methods for the discovery and enhance the accuracy of the result, so as to reduce the cost and time. So if you look at whether you ask me whether we can use a full bioinformatic approaches or combining the traditional and bioinformatics, I would say it depends on your design. So if you have a budget Of course, sometimes you can go all the way to both approaches. And let's say you have the technical skills or know-how in bioinformatics, you can do all full-fledged with the bioinformatic approaches. I think that's all from my presentation today. So these are the references. So I think, thank you for your attention.
Thank you, Professor Dr. Shaquin. Please give a round of applause. So after we have Professor Dr. Shaquin about how bioinformatics have an important role in drug discovery, I will open sections for questions for all the audience, for the students, for the colleagues, and all the professors that we've been here today. Please, if you have a question, please raise your hand. You might ask a question in Indonesia, and we will deliver it to Professor Dr. Shaquin. Please. Maybe we can have some question in the chat page as well. No? From the students, please. Or maybe for a question from myself. So we have listened from you how bioinformatics have changed the way we discover drugs in recent days. From the traditional way to bioinformatics way, there are a lot of changes. And we have students here and also college in biomedical. My question is, what is the important knowledge and skill that all students should have so they can be involved in that area as well in the future, maybe for the future career as well? Thank you.
For the question, very interesting question. I think now I think it's the era of bioinformatics and also informatics, as you say, that digital era. So like it or not, I think it's not only biological subjects or field, it venture into all type of fields, name it, even in finance, in social. So you cannot escape by incorporation of digital knowledge. So I think it is wise to actually equip our students with all this bioinformatic knowledge. So now maybe you may ask me how deep that a person have to be involved in bioinformatics. Of course, there's no limit. So I would say, however, knowing some of the basic search in bioinformatics using a certain databases, so that will also facilitate the understanding on the fundamental of science and also biology, and whatnot in the biomedical and in the treatment and also diagnostic, etc. So yes, I think most of these biomedical science program have incorporated the bioinformatics as part of their curriculum. And I think students wise, I will advise you, you should pick up all this knowledge along the line. If it's not drug discovery, it also helps you in other means of studies, like for example, big data, machine learnings. So those are the things that will be very helpful when you are out to the working world. Yeah, thanks.
Thank you, Professor. So untuk mahasiswa-mahasiswa dari program terutama S1 Biomedis, seperti yang telah disampaikan oleh Prof, bahwa bagaimana bioinformatik ini akan menjadi hal yang sangat penting di masa depan, dan tentunya adik-adik ini adalah hal yang mungkin baru bagi adik-adik dan akan sangat menjadi menarik jika adik-adik bisa menjadi orang-orang yang ahli di bidang tersebut. Masa depan akan sangat terbuka sekali. Jika bukan hanya drug discovery ya, Profesor, tetapi juga apakah itu machine learning, juga pengolahan dengan big data lainnya, itu akan menjadi hal yang sangat menarik dan penting di masa depan. Dan peran-peran adik-adik tentunya juga otomatis akan sangat penting dan diharapkan. Nah, seperti itu. Oke, From the student, please. Maybe. Okay, very good.
Okay, thank you, Prof. Okay, in this occasion, I have a question for you. Let's introduce myself. My name is Agnes. So I have a question for you about molecular docking. I'm very interested about the molecular docking, and so my question is, what are the limitations of molecular docking, or what are the problem of molecular docking if we use the molecular docking for bioinformatica in drug delivery. Oh yeah, drug discovery this type. Okay, thank you, sir.
Thank you, Agnes, for the questions. Terima kasih. So now, saya dipahamkan bahawa ramai di antara yang involved dalam ujian bahan ini untuk drug, dia uses docking, betul? So yes, there's a limitation. And also, as I mentioned, keep mentioning and over again, there's no ideal. Dia tak ada satu cara yang sangat sempurna. So setiap caranya adanya kelemahan dan kebaikan setentunya. So of course, docking itself, first and foremost, you need the structures. So kalau you tak dapat structure yang betul, so your docking won't be accurate. So it all depends on what you key in and upload into the systems, correct? So and secondly, it's just a simulation. Sometimes when you dock correctly, doesn't mean that in biological, it happens what you actually simulate. So that's why apart from that, docking also you need several approaches in terms of dock. Of course, it's a very long details here I won't be able to share. So now when you do a docking, as I said, the structures might be correct. So if let's say if it's incorrect, you assume it's correct sometimes. So that will be something disaster when it comes to synthesizing the real particular compounds. Yeah. So because this limitation is the system is only read what you actually input. And also, of course, it also look at what are the available targets there in the database. So let's say you are looking for certain targets that are not available or certain molecules, then will be something not comprehensive enough. Dia tak secukupnya untuk screening. Yeah.
Terima kasih, Profesor. Bagaimana, Agnes? Apakah masih ada yang ingin ditanyakan? Okay, please, another student.
Thank you for the opportunity, sir. Let me introduce myself. My name is Kecha Putri Febiona, and I would like to ask, in drug discovery, some of the process, one of the process is clinical trials, right, sir? So I like to ask, if it's possible in the future, if we just use simulation in clinical trials with animals, because there is some people that disagree if we use real animals because they think that's animal cruelty. If it possible, if we just use simulation in computer for clinical trials, what is the disadvantage if we just use simulation for clinical trials, sir? Thank you for the opportunity.
Very interesting subject and topic about whether you can use bioinformatic to skip the animal trials and then straight go to the human clinical trials. So the clinical trials here is not necessary, I'm saying not the human trials, because I didn't put it human clinical trials. The clinical trials can be animal clinical trials as well. So what happened is when you use this bioinformatic approaches, doesn't mean that you can skip all the animal trials, right? Really? So you understand that means it's not that after bioinformatic, after do all the simulations, straight away you go to human clinical. That's not. It's just that by using this high throughput screening, so you'll be able to save or reduce the use of number of animals. Okay. So, and of course, with that, it will give you a better insight of the interaction of the molecules towards overview of the molecules inside the animals or inside that particular organisms. So it is not just to skip, but it's reducing the use of animals. And that will actually helps in terms of sacrificing unnecessarily. Okay. So of course, the route is still the same. You have to really go through the in vitro, in vivo, and then animal in vivo, and also human clinical trials, because these are all some requirements by some agency like FDA, etc., WHO, etc. Yeah.
Thank you, sir.
Yeah.
Thanks. Yes. Very good questions from our students. Thank you so much, Professor, for the explanations. Is there any other students who want to give a questions to Professor Cha? From the audiences who join us via online, do we have a questions? Please. No. Okay. Mungkin terakhir, Profesor. Di sini mungkin kita sudah banyak mendengar tadi mengenai peran dari bioinformatics. Nah, saya melihat di sini Profesor juga bukan hanya sebagai seorang saintis yang sudah sangat diakui di seluruh dunia, tapi juga saya kagum dengan Profesor juga terlibat secara aktif di fakultas maupun universitas. Program ini kan juga adalah bagian dari program Universitas Andalas untuk mengupayakan kami juga akan menjadi menuju world class university. How do you suggest our faculty as well as university, which one will be our priority? It is the teachers' development, it is the students, or the outcomes of the students, or the systems, or anything that, according to your experience, that we have to prioritize to make our university become the world-class university? Okay.
Wow, these are very heavy questions. So I would say that in all aspects, I would never say that one particular aspects that cover everything. So as you know, educations, students is all our products, you see. So, and also the curriculum is something that in place to enrich them with proper skills and knowledge. So that's also something, the advancement of the particular curriculum is very important. So to make sure that this curriculum has been reviewed and also up to date to the tradition, to the trends of the current needs. For example, in digital, so you should include all these digital component in the curriculum, and depends on what that required by the particular profession or the bodies. If there's any bodies that governs this particular profession. So of course, not forgetting also the staff need to advance themselves in all those areas that is highly demanded now. So as you know that now in the era of AI, artificial intelligence as well, so many people also worried some of the profession can be taken over by AI. I think yesterday we have a short discussion on that. So not to worry, because as we know that some of the skills, no matter how, is still not able to 100% taken over by AI. So that's, you need to make sure that your curriculum have embedded a certain component skills training that will not be 100% taken over by the AI in the future. So this will secure that particular student who graduated from this particular system. And you will ask me about Fakultas and also the university. I would say that personally is really good, because when I visited the lab, I say that most of the labs actually are well equipped. So I think you can go even faster with this networking and also interaction with others component. As you know that in research, we are working in a group, and also if you wanted to make yourself seen, and then you need to collaborate with more people externally or in between different universities in the countries, and to make sure that it becomes stronger at a point where the basic of this research become more renowned in the world. Yeah. I hope that my answers actually give some of this input to you. Thanks. Yeah.
Thank you very much, Professor. It is a very insightful suggest for us, and we hope that our program today is another step for us to collaborate and to develop our faculty to become a world-class faculty as well. Thank you very much, sir. Okay. Thank you. That's all for our seminar sections today. Thank you to our respected speaker, Professor Dr. Maybe we want our seminar here. At this moment, we still have one interesting question. May I, Professor? We have another question. Okay. Yeah, the question is from Dr. Teguh Yuki Ananda. Thank you for your kindness, Prof. I would like to ask, what could be the biggest problems in bioinformatics role in drug discovery? So maybe the challenges about bioinformatics in drug discovery. Please, Professor.
Yeah, thanks, Dr. Tengku, for your questions. Very related questions to the topic. So what is the biggest problem? So now in drug discovery, as you know, in bioinformatics, okay, it is a simulation. So all we know that sometimes simulation and all these things is not 100% that it will realize in the biological world. So now what happened is the other component of problems will be the technology know-how. So if let's say once researchers and also doesn't have enough capacity of understanding the bioinformatics component in drugs discovery and lack of certain information that is to be incorporated in the particular screening, then that will actually lead to some of this misinformation. So during this particular prediction and also simulation. So that is something that we need to understand. And of course, most often that the connections, so and also the availabilities of the infrastructures. So some of the countries, like it or not, or area or organization would not capable have the infrastructures in place to support the bioinformatics screenings, etc. So of course, you know that it's easy to say than doing it, you see. So because some of these particular software may need a different capacity in terms of screening and also predicting a certain compounds, etc. So that is also part of the limitation of it. Okay, thanks. I think there's another one.
Thank you, Professor. Maybe we can have another one. Yeah, from Ika. Dear Professor, thank you for your excellent presentations and very interesting topic, actually. I would like to know what kind of troubleshooting do you use when the concentrations of samples on the ELISA too low, far away the minimum of the Q standard. Is there any clue to able to analyze the data instead of repeating the experiments again? Best regard. Maybe this is very practical questions, but I think that's interesting as well. Please, Professor.
Oh, okay. Thanks, Dr. Ika, for your question. This question should be very specific. I think it's on the concentration of the sample or in ELISA too low. I would like to know what kind of troubleshooting do you use when the concentration of the sample of the ELISA too low, far away from the minimum of the sample. I didn't quite understand the questions. What does it mean by concentration of the samples too low? Of course, you don't have enough samples, of course, how to run, right? You cannot, yeah, some of the experiment you cannot do wonders because dia bukan magic. So dia tak, you letak satu microlith, then it comes out magic, suddenly the results come out. Of course, if it's sample dependent punya technique, so like it or not, you need to get more samples, right? You cannot be saying that with this limited data, then suddenly it turns out tons of results. Tak boleh, So I don't think so, because these are all very wet lab-based orientation. Because if you use bioinformatics, of course, bioinformatics doesn't have concentration. So they're just data that put in, correct? But you're talking wet lab like ELISA and then PCR and all these things. So that one is very subjected to the initial samples that you use. After you have run it, but you make if there's no way that you can actually analyze, even through the bioinformatic approaches, you won't be able to solve these issues. You just need to rerun with a appropriate samples concentration to make sure that the result that you obtain is valid. Dia tak boleh just because of a projection. So I'm trying to understand whether Dr. Ika to say that it's projection, okay? So tak boleh, dia tak boleh just project. I mean, extrapolate your graph and then to say that it's actually predicted. That is not really a good in terms of the science.
So there is no way then to get appropriate samples. Yeah, Professor. Okay. Thank you very much, Professor Dr. Cha. Another questions? Okay. No. Alhamdulillah, thank you very much, our honored speaker, Professor Dr. Shah, your queen, who have joined us today in a very special moment in the seminar. And thank you for all our professor, colleagues, staffs, and students who have joined us today live at the faculty and via online as well. Thank you very much. And maybe we could end our seminars today, and I will give back the time to our MC, to Mrs. Sinta, please. Thank you very much.
Bapak dan Ibu para undangan yang saya hormati, sebelum kita akhiri acara ini, kami persilakan kepada Ibu Ketua Program Studi S1 Biomedis memberikan kenang-kenangan dari Fakultas Kedokteran Universitas Andalas kepada narasumber kita, Profesor Dr. Kedokteran, PhD. Kami persilakan. Dilanjutkan dengan foto bersama. Kami persilakan. Kami persilakan kepada staf pengajar beserta mahasiswa untuk bisa berfoto bersama dengan narasumber kita. Kami persilakan. Kami persilakan kepada mahasiswa untuk berada di atas podium. Kelihatan kan sana Pak ya. Lagi. Direkam. Video. Belajar dulu, belajar. Oke. Tuh. Oke, kita mulai serentak ya. 1, 2, 3. Dah. Tapi tadi kayaknya nggak jelas gitu ngomongnya. Bisa lebih di ini lagi nggak ya? Kompak lagi deh. Oke ya. Sekali lagi ya kita coba ya. Kita coba sekali lagi. Tapi ini ya, belum yang fitsnya. Kita coba sekali lagi. Oke. 1, 2, 3. Sip. Oke. Kayaknya cukup deh. Nggak ulang lagi. Oh, masih ulang Ibu? Sekali lagi. Enggak, yang tadi ini yang fitsnya ya, yang oke-nya ya Bu ya. Berarti ini yang terakhir, tolong kata-katanya yang bagus. Oke Dek, siap? Ini yang barisan belakang mana nih, nggak kelihatan kepalanya? Oke, sip ya. 1, 2, 3. Oke, beri sudah seluruh rangkaian acara kita pada hari ini. Terima kasih atas kehadiran dan partisipasi dari Bapak Ibu serta tamu undangan yang telah hadir pada hari ini. Mohon maaf atas segala kekurangan. Saya akhiri dengan wabillahi taufik wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Jadi dong.